would be when another co-specific is listening for sounds that are not meant for them. And you can also have things like kleptoparasitism, which is this bat finds a prey through sonar, but there is another one of his species that's listening, and it will get to the prey before the one that actually had the trouble finding. And those are just some examples. And so when we are studying sounds, we are, as primates, we are actually very visual. So what we do most of the times is to transform sound in an image to be able to study that sound. And we do that normally through spectrograms. Spectrograms are three-dimensional images of sounds that have a frequency axis and a time axis. And you have also a color coding that indicates the intensity. So we have here an example of a bat echolocation. And you can see that the most bright colors are, in this case, represent the most intense sounds. And this is Myotis bat. And just for you to start understanding a little bit of bat bioacoustics, this is what we call a pass. That is, the bat is passing. And this part here, it's called a feeding buzz. It's the moment where the bat gets closer to its prey and we will finally attempt to capture it. So through this part, you can see a specific activity that it's doing. This is a feeding attempt. So just through sound, you can ask several questions. And in this case, where you have a sound that indicates a behavior, you can, for instance, ask if it is feeding more, it makes more feeding attempts on different conditions. In this case, in the example I'm giving you, we are looking for moments where the light conditions are different. So we look to moon phases and sunset and sunrise, because in those twilight moments you have, of course, more light and the bats are active. So how do we conduct this research? How do we answer this kind of question, this specific question, with bioacoustics? You go somewhere. In this case, it was Nízia Floresta, here in Rio Grande do Norte state. You go somewhere, you install a recorder, and you go there many times to change batteries and memory cards, and you get a ton of recordings. Those are recordings of 4 different families of bats that we found. and those are the feeding buzzes of each family. And you go and you analyze the recordings by counting passes and counting what we found in this case is that here are the passes. There are difference in the number of passes between twilight and dark, but not full moon, but not feeding buzzes. So you analyze, do the statistics, and you get your answer. That is, well, light does affect passes, but not feeding attempts. And this is just one quick example of the kind of research that we do on our lab. And I'll bring another example from tiny bats to very large whales. This is another work we have done with humpback whales. In this case, what we had was recordings of humpback whale songs in the state of Maranhão. And we wanted to know from where had this whale came. Because the population that breeds on the Brazilian coast is called the breeding stock A, but typically they are more concentrated in the most southern parts of Brazil, Bahia and Rio de Janeiro, and less in smaller latitudes. So we had a whale singing very far from the traditional population area, and we wanted to know if it came from the same region, from the same population. And in the case of humpback whales, what happens is that in every breeding season, all the males of that population are singing the same song. So we compared the recordings and we found a match between the song units of recordings in a more central part of the breeding population and the recordings that we had that were far away. So yes, the whales we recorded here were in fact from the breeding stock A. So those are two examples of what we can do with bioacoustics and what kind of research we have conducted. And now I'm going to start the part that I really love to talk about, which is maned wolves. Maned wolves are not traditionally the best model to study with bioacoustics, because unlike cetaceans that do everything in their lives are sound, they do it to reproduce, to navigate, and many social interactions. And bats also use sounds for everything, but not large terrestrial mammals, because it's usually assumed they are not that vocally active, and they mostly interact physically in contact with each other. So it's not a very classical model. So why study maned wolves with bioacoustics? First, they are very difficult to study because they are nocturnal, they are very shy, and they are solitary. Unlike other big canines, they do not form packs. Cooperative hunting, it's very rare. And the breeding pair, they are monogamic and both parents care for the youngs, but the breeding pair do not stay together all the year. They only meet for breeding. So it's kind of a very solitary species. And because of that, they occur also in very low densities because they have very, very large home ranges. And the pair do not defend it simultaneously. At least that's not what people thought. So if it's so difficult to study this species, maybe you could monitor it through its sounds, because it uses chemical, of course, but it also would use acoustic marking for many functions that were not that very elucidated at the time, and both sexes use it. So our question was, how do men and wolves interact acoustically? This is the larger question, but we of course have to break it in small pieces. So I'm really hoping you'll be able to listen to it. Okay. How can we? Okay. This is the roar bark. This is the main man and wolf vocalization, and you can hear, I hope, it's really loud, and so it's a very good candidate to monitoring projects. But first, we needed some basic information. And what basic information are those? Which sounds do men and wolf make? How often? And what information is being conveyed? And to answer those more basic questions, we went to captivity facilities, and in this case, Victor Sabato made a lot of recordings. He spent some more than 60 nights watching and recording the men and wolves. And what he found is that the man and wolf has a very large set of different sounds it makes. And this is called a repertoire. And it goes from a more close-ranged vocalization like whines and growls and grunts to more long-range vocalizations like roar barks and the single bark. And all those vocalizations was a surprise, because there is a theory that proposes that more complex, socially complex species have more complex and bigger repertoires, which meant that maybe the man wolf has a more complex social life than we thought. Now, we'll focus specifically on the raw barks because it emits, then it's a long-range vocalization. They emit it reasonably often. And another thing that we found is that it emits more near mating. And one very nice finding was that one of the information it conveys is identity and sex. And I'll play you an example, and I want you to try to notice the difference between the different individuals in the recording. And if you are curious, the QR code it's for SoundCloud with some examples of other maned wolves vocalizations, this one and others examples. So you can go check it out. So now we go to the wild. We conducted our research in the Serra da Canastra National Park in Minas Gerais state. And we wanted to answer the questions of if we could monitor free-ranging maned wolf through their sounds, and how often did they communicate in the wild, what is the function of raw bark, and especially with whom are they communicating. So we went to the Serra da Canastra National Park, and we installed a bunch of autonomous recorders. Those are equipments that you place in the field and you program it to record. It may be continuously, it may be, in our case, it was only at night, and so on. And you leave it recording automatically in the wild. Of course, you have to change batteries from time to time, but it does most of the work for you. So we installed a bunch of autonomous recorders, and we left them active for recording something like 291 nights, almost 300 nights of recordings. And then we analyzed the spectrograms, and through a software that automatically finds the raw barks, we developed that automatic finder, and we got a lot of raw bark sequence. So in total, we got 1,321 raw bark sequence with more than 14,000 raw barks. And this is an example. They make those sequence of about 10 raw barks. This is just a clip, an example with 5 raw barks, and I'll play for you. those are wild recordings, so the sound is much softer, but we could record many sequences, which mean, yes, it is possible to monitor free-ranging man-eared wolves through sounds. And one of the most interesting things we found were those sequences. And I'll play for you so you can maybe notice something different. As you have guessed, this is a raw bark sequence with two animals. You have this one and this other one that alternates raw barks. You can see that this one has a certain cadence of his vocalization, and the other one here is also very clear that there are two animals in this case. They have some intensity difference, but also some instance where there are the raw barks are overlaid, and that indicates for sure that there are more than one animal. So we found around 12% of the sequences that we found had more than one animal. Always, most of the times, two animals. And that was really interesting. And we then came with our next question, because science is made that way. You answer one question, and it gives you 2 or 3 more questions, and you go and make studies to answer those, and more questions arise, and so forth. But then we wanted to know which were the animals that were interacting acoustically. And the first question to answer that was, are the animals that are vocalizing, are they close? Meaning, are they together, close by each other when they vocalize? And we made other analysis with the same recording data set, and for some sequences, raw bark sequences, we could estimate the distance between the participants. And what we found was that on average, they are more than 400 meters apart. And sometimes they are really far, something like 1.5 kilometers apart from each other. So no, they are definitely not close by. Usually, they are not close by when they vocalize. So they are, each is one region, and they are vocalizing far away from each other. Then the next question that help us understand the man-eared wolf acoustic communication is, when do they produce those raw bark sequences? And this is throughout the year, and we have here a lot of raw bark sequences during the mating period, just like we found in captivity. And also there are some other rollback sequences near the birth of the pups. So this indicates that rollbacks are being used to reproductive reasons, to attract mates, and also to coordinate parental care. But they also do rollback sequence in the non-reproductive season, which means it's probably also, it probably functions as a territorial announcement and territorial defense. But when you have, when you see only the sequences in which there were more than one animal interacting at the same time, we can see that they rarely produce those sequences in the non-reproductive season. It's mostly in the reproductive season, which indicates it has something to do with reproduction, of course. So still answering when, you can look at other cycles other than annual. You can see lunar cycles. And what we found was that maned wolves produce more rollback sequence near the full moon, which means better illuminated nights make them more chatty. But not for the group sequence, no pattern found in the group sequence that indicates that for the group sequences, there are other factors that are influencing this kind of sequence. But for the sequence with only one animal, this means that maybe on better illuminated nights, they can see their prey better, and they have better feeding. They feed faster, and so they have more time to vocalize. And though the prey may also see them better in better illuminated nights, which normally mean they also avoid the predators. But I need to remind you that the maned wolf is actually omnivore. It feeds on fruits also and vegetable eatings. That means that better illuminated nights may help them find fruits, and they may have more time to allocate in communication. Finally, third part of answer when, you can see at dial patterns. And what we found is that it is crepuscular, especially in the dusk and the first half of the night. It's very vocally active, and less so in the second part of the night, and almost nothing during the day. This brown line represents the activity pattern that was measured through radio collars with telemetry. So it's a pattern of activity and rest, real pattern of activity and rest. And you may see that it does follow reasonably well the vocalizations, but when it gets to sunrise, to dawn, there's a peak in activity, but it is not followed by a peak in vocal activity. And naturally, one of our new question was, why is that? Why it is active at down but not vocally active? And also, if group vocalizations followed a similar pattern as normal robark sequence. And just like the moon, there's no pattern in group vocalizations, no dial pattern, which indicate that when it's a group vocalization, they have to answer no matter what the hour, no matter what the moon phase, illumination, or anything. This is a kind of vocalization that maybe functions for a slightly different purpose. But normal vocalizations follow this dial pattern. Now, to answer that question of why do they concentrate their activity in the dusk and the first half of the night and not the second half or dawn, we conducted a playback experiment. And a playback is when you get a recording of the animal, you go to the field, and you play that recording to the animal, and what you're going to analyze is the animal response to that sound. You are basically asking the animal a question through their own vocalizations. So, is there a reason why dusk? What we thought was maybe robarks propagate better during dusk, during those periods, and that's why it vocalizes more during that time. And so we conducted the playback not only to see if the man-eared wolf responded and how they responded, but also to re-record with other recorders, re-record the same sound and see if we could find a difference in propagation. So here I am in the field with the loudspeaker, and this is the first recorder. We positioned other recorders at different distance, 20, 40, 80, and so on meters, in 3 different places. And then we got the recordings and we analyzed the recordings, measuring some things like how much of the intensity it loses with distance. For those of you that are interested in the physics of this part, the more mathematical calculations, bioacoustics is a very nice field for that, because you can really enter in very complex calculations. I will not do that today, but just if you are interested in that part, bioacoustics is for you, also for you. So you have here an example of a spectrogram of a raw bark, and the same raw bark recorded in different distance, and you can see it degrades over time, over distance actually. So is dusk a better time to vocalize? Here we have the data separated by the hours that we conducted the playback. And well, what we found was that dusk is actually a good time to vocalize. So it's the first half of the night. But 5 a.m., that's dawn, the sunrise, is the best moment to vocalize. It's the moment where vocalizations better propagate. And so we still haven't answered we still haven't answered why do they prefer to vocalize in the beginning of the night and not in the end of the night. One of our guess is that it's not of a physics-related matter. It's actually because they have to announce in the start of their activity that they are alive and that range is theirs. The range is occupied. Something very similar to birds that vocalize during dawn, because that's their start of activity in the morning, and they need to announce straightforward right in the beginning of the activity that they have survived the night. In the case of the man and wolf, that they have survived the day and they are there to defend their territory. We haven't answered the matter of the dusk, but we have some very interesting finding regarding the behavioral response men and wolves had to our playback. We conducted it in different moments, hours of the day, and we had a lot of responses, many responses, but only near dusk. We had no response in any other time we conducted the playback, which is another clue that it might be something like defense mechanism, something to defend territories and mates. And one very interesting thing was that we had some group responses. They did not intercalated the raw barks with our playback. They intercalated entire sequences. So we did the playback, we waited, they would respond with a sequence, then we would make another playback, they would stop calling, and then we would wait, and they would respond again after the end of our sequence. So when it seems that this intercalation of raw barks, it's maybe not for territorial defense, maybe for a more specific function. And what was interesting was that two animals approached our playback, but unfortunately we could not see them because it was night, and the maned wolves managed to flee from us, and we could not see if, for instance, if it was a male and a female. Maybe we could identify the specific maned wolf because some of them we knew at the time, some of them, but none of that was possible, unfortunately. So finally, we answered a question using all three kinds of methodology that we used so far. The recordings in captivity, the passive recordings in the wild, that is just placing the recorders and doing the recordings, and the playback. And we wanted to use all three data sets to answer how do maned wolves interact and which maned wolves are interacting. So to answer that, again, you have to break in a lot of questions, smaller questions. So our first question is, do the interacting maned wolves differ in their vocalizations? And do they differ? Who are interacting? In captivity, we had this answer. You can see that they do differ a lot acoustically, and you can, as I have said, we found that you can discriminate sex and identity through their vocalizations, and of course, at that time, Victor was seeing the animals. He knew which animals which were vocalizing, and what he found in captivity is that almost always, with just one exception, one single sequence, all the other sequences were between males and females. Sometimes because in captivity, animals are closer together. You have two animals in this enclosure, and then you have two animals in a very close by enclosure, which means there were a lot of sequences with more than three animals. But in all the cases, except one, it involved males and females. It was multi-sex vocalizations. And could we do something like that for the wild recordings? Can we discriminate which maned wolves are interacting? And well, first we needed to use the propagation data to see if the acoustic parameters that are used to discriminate identity and sex, if they are stable through distance, because otherwise we could find our results would find that the interacting maned wolf differ, but maybe it was just a matter of the distance to the recorder, if the acoustic parameters were not stable. So we conducted a study to see if they were stable. And yes, some of them are stable for a certain distance. So we just got the sequences that were in a safe distance that had a safe intensity to measure acoustic parameters, and only the acoustic parameters that were stable. And we did measure the difference between wild maned wolves that were interacting acoustically. Let me play for you so you can try to listen if you can notice some difference between the two maned wolves. We did the measurements, and what we found was that when comparing the two maned wolves that were vocally interacting, that there were always one that had a shorter raw bark and one that had a longest raw bark, and this difference is statistically significant. And also there were a difference in frequency. Females were higher in frequency, that is, they were higher pitched, and males, sorry, that was in captivity. Between the interacting maned wolf, there was always one that were higher in frequency and one lower in frequency, and this difference is also statistically significant. So you have always one with lower frequency and one with higher frequency. And we then compare this with what we had found in captivity. And in captivity, there's a significant difference between male and females in the duration of the raw bark. The male raw bark is longer and the female is shorter. And you have a tendency, not statistically significant in captivity, but we have a tendency for females to be higher in frequency and males to be lower in frequency, which makes sense because of their size. Males are slightly larger than females, and thus it makes sense that they would produce slightly lower frequency vocalizations. So when we take everything together, we can conclude that at least most of the time, the two interacting maned wolves in the raw bark sequence are a male and a female. And that was really nice to find, because that it probably indicates that this is a vocalization mostly used in between breeding pairs, because they form stable monogamic pairs. They are not together, but they are often in contact with each other, so it's a kind of long distance relationship. So we have answered a lot of questions to resume we know that man-eared wolves do interact a lot, much more than we had thought. That sequence with only one animal are emitted throughout the year. That dusk and the first half of the nights are very important, probably because of territorial announcement function of the roebucks, that the interacting man-eared wolves are not close. We have many group sequences in the reproductive season, and group sequences are mostly male and female interactions, which indicates that it is the reproductive pair, and it could be for them to mediate their meeting during the breeding season, can be an intra-pair display, can play a role in mate guarding, and also in parental care coordination, because if they are not together, close by at least, they need a way to manage all those behaviors. And we propose that they do that through sounds. And with that, I'm ending my talk. I hope I have helped you understand a little bit of the multitude of things you can do with bioacoustics, especially to answer behavioral questions, but you could also use it to answer cognitive questions, but that's not my specialty, so this talk was mostly behavioral. And I have a lot of people to thank, and you can you can check our website, our IG, and you can also see here many more raw bark sequences in my SoundCloud, that's with this QR code or with this link. And with that, I would like to thank the organizing committee and also everyone that's watching. And now, if you have any question, I'll be happy to answer.
I have a question.
Okay.
Yes, I am very impressed with your work, truly.
Oh, thank you.
In particular with the lunar cycles in the main wolves.
Nice.
Yes, but it's a simple question before pass to this event of the academy. Okay, meet the plenary speaker. But do you can contrast the data of the lunar cycles with I don't know, luminic contamination?
Oh no, but that would be very nice to do. We have when you say the luminic contamination, you're saying artificial lightning or aluminium. I didn't heard.
Yes, artificial lighting, because I have an interest in these studies of behavior, in particular with other animals, but behaviors and how they use the polarized light to do
really nice
stuff, no? Yes. The polarized light of the moon, no? Because the artificial light interferes with that.
Oh, yes. In this particular case, it's actually a very kind of isolated preserve. So there's the region that we recorded has no artificial light whatsoever. But in the bat example I gave, one of the places we recorded is highly light polluted. So maybe one of the reasons we didn't found a relationship with full moon and new moon, maybe it was because of light pollution. So in short,
Well,
not with men and wolves, but yes, with bats.
okay. Well, thank you. And only because we are on time, I don't know. Let me only check. To meet the plenary, the activity, we'll start with the next question. It's difficult to you to introduce you to the world of academy because you do a lot of work, field work, no? You do a lot of field work. And it's common to, as you say, the field work, you don't have opportunity to insert it in the academy. Yes, it was difficult. I think some people would like to hear about that.
Not really, actually, because I don't think it's usually that way, but I was very lucky to find a lab that was very, very connected, very, it's called the ear hub. Hub is for, it really functions as a hub. So this highly connected lab made my way very, very easy to do both the field work and the lab work, return to the academy. So I don't know, my experience was very, very positive, and I invite anyone that has interest in bioacoustics to join the lab of bioacoustics, the ear hub, and maybe they will have the same facility that I had.
And only for clarify, when you were doing all these studies, your focus, for example, a year, 2 years of your life to only do field work and keep the data, and after how many time do you spend in working in the articles or the office investigation?
Well, starting with the field work, it's kind of reasonably easy. Maybe it's not always like that, but in our case, the recorders, you leave the recorders there, so, and you get the data from time to time. So I had more time with the data, but once I have the data, it takes a lot of time to process. It takes around the same amount, and that's like going for the low estimation. You usually take the same amount of time of the recording, at least that, to analyze. So if I have 291 nights of recordings, I spent that amount of days analyzing it, and then and for publishing, actually writing the paper and publishing, I would take 1 month to actually write the paper, and then it goes to all the co-authors, and they spend, I don't know, something like another month revising it, and then we submit, and that's I don't know, some to 6 months before it can be published.
Okay, as a full semester before publish. We have a participation of another plenary speaker, Laura Hernandez. After her participation, we are going to a break and we are going to come back. But only let me, can you turn on your microphone? Yes.
Can you hear me? Yes. Everything is, yes. Thank you very much. Thank you, Camilo. Thank you very much for your talk. I mean, when always that I'm listen someone to talk about acoustics, like excitedly, because currently I'm working on acoustics in dolphins. But I think acoustics in general, it's very interesting because it's something that we don't like. Sometimes we don't know, or it's not easy to interpret the activity, right? And well, what I want to ask you is, well, you like compare the activity like intra-species interaction, right? In different seasons with the lunar cycle. But have you considered now that with this information, you can like add the another variable, like for example, the soundscape, like sound, like maybe if the soundscape have an effect on this kind of intra-species interaction, because in most of the animals, I mean, I think that in 99% it works like that, no? I mean, the soundscapes have an effect on how they interact between them, not aside from another species, between them in the group.
Yes, I have two line of works that can kind of answer your question. One is that yes, we have a work, but it's very it's on its beginning, right? Analyzing this kind of Serra da Canastra soundscape to see where the maned wolf inserts itself in the soundscape. And one of the things that we have found, let me see if no, I have to upload. Let me present again. Okay, so if you see the wild recordings here, you can notice that it has this band which is occupied by insects. Those are probably grasshopper. And you can see that the maned wolf is exactly just below. So this is a more silent band, and the maned wolf seems to use precisely this band to communicate. And then you have insects that are in higher frequencies, and they do make, in this case, it's hard to see because the recordings were only up to 4 kilohertz, but there are a lot of bands of different insects. And also, I don't think there are an example here, but there are anurans that vocalize below the maned wolf. So we have this acoustic niche partitioning, and it was really nice finding, but this work it's only only on its beginning. So I don't have anything very conclusive to say. But yes, they do different animals interacting in the same soundscape. They seem to have this partitioning. They see it seems to be relevant. The other work that might relate to your question, it's a work we conducted with the monitoring of dogs, feral dogs.
Ooh.
So we monitor simultaneously. We monitor the maned wolf through its paw barks, and we also monitor the dogs inside the park. They should not be there, okay, just to be clear. Inside the park, we monitor the dogs through their barks, and we want to see if there is a relationship between wolves and dogs. Maybe the maned wolves avoid the areas or times that dogs are barking. Maybe it's a kind of risk ecology and risk avoidance ecology. But this work is also kind of preliminary, so I also do not have any.
It's interesting that you mentioned that the feral dogs, because maybe in this kind of situations, they like compete for the resources, because it's like the eye droppings, eye droppings. No, I mean the feral dogs have learned like the moment or the sign, the vocalization for
the
a
maned wolf.
particular behavior. And okay, if they are vocalization like that, it means that it's because it's a predator, or they have found food or something like that. No, so it's very interesting. Wow. Well, I expect that you have the 100% success in your work in this.
Oh, thank you.
Your research, really, because it's pretty interesting. And with feral dogs, that like it's, I think that it's a global problem nowadays, like with the protection of the wildlife. So good luck. No,
Thank you.
thank you very much.
So we are going to a break of half hour, and we are come back. Even for the people that is looking us in YouTube and the new guest. The transmission is not going to end, and I'm going only to put the app again, and see you in half an hour. Thank you for
Okay.
your time now.
Thank you all.
See you in a moment. Hello, everyone. We are going to come back in 10 minutes. Now I'm going to check some stuff before start and see you in 10 minutes. Hello, everyone. We are going to come back to the second day of International Student Conference of Animal Behavior and Cognition. I'm going to present, to introduce you to our first exponent, Deyanira Aranda from Mexico. And let me add to this call.
Hello, can you hear me?
Yes, I can hear you. We can hear you.
Great. My name is Deyanira. I was with you yesterday during Maths plenary, and I am really happy to be back here. So I am going to start. I am going to try to present it. Let's share the screen. So can you see the presentation now? Hi, can someone actually tell me if you can see the... Camilo?
Sorry, my microphone is turned off. Yes, we are transmitting on YouTube and we can see you and hear you.
Great, thank you. So today I am going to talk about a physics concept in guppies. It's the Brace paradox. One moment. Okay, so this study was conducted by me, Deyanira Aranda, with the help of the biologist Eduardo López Sosa and Dr. Palestina Guevara Fiore and Dr. Felipe Pacheco Vázquez. Sorry, Camilo, please turn off your microphone.
Yes. Sorry.
So yes, Brace paradox is a phenomenon in network theory where adding extra capacity can paradoxically worsen overall performance by increasing congestion. This occurs because individuals acting in self-interest shift routes to use a new option, leading to inefficiencies and bottlenecks. The paradox highlights that more options don't always improve flow and can sometimes have the opposite effect. For example, imagine you're in a city with two main roads and traffic flows smoothly. Then the city builds a new shortcut between these two roads. You'd think this shortcut would help people reach their destinations faster, but in many cases, more people use this new road, causing traffic jams. Ultimately, everyone might spend more time on the road than before the shortcut was built. This is really, you can see this a lot when you have to, when there is like a road that gets closed, and then you try to find a new route and you start like moving to other sites, and suddenly everywhere is a traffic jam. That's basically what happens with a Brace paradox. Since Dietrich Brace described this paradox in 1968, it has been studied in various contexts like traffic systems, computer networks, mechanical networks, electrical circuits, and blood vessels. Our goal was to explore whether this paradox applies in a biological system, specifically in the movement of guppies. We are interested in how animals make decisions in constrained environments and whether adding more paths leads to more efficient movement or to more congestion. But why did we choose this system? That's because interdisciplinary research, in this case, physics and biology, enables us to integrate a biological perspective into inherently physical theories, thereby testing the universality of this phenomena. We decided to use guppies because they are widely used in collective dynamics and biological systems, especially in the studies of collective behavior, They are small in size, they are easy to handle, and they are cognitively flexible, which allows us to explore how environmental factors like different path options or the presence of threats affect our decision-making and group movement. Guppies can also integrate both prior experience and information to optimize decision-making, which makes them ideal for dynamic decision-making testing. They also move in 3D and adjust their behavior based on group context, which mirrors some aspects of more complex network behavior. It's important to distinguish between shoaling and schooling. Shoaling is a loose gathering of fish that stay together for social reasons without a strict synchronization. This could be, for example, when we go out with friends, we go to the same places, we might interact with each other, but we do not move at the same time. While schooling is a tightly coordinated group where fish swim in unison. The example that I can remember is the fish that forms like faces in the movie Finding Nemo. They are a perfect example of a schooling fish. And we decided to use only female guppies for these tests because female guppies form more cohesive groups than males, with stronger social bonds and tighter coordination within their shoals. Guppies often act collectively and make decisions collectively, especially in a social contexts like foraging or avoiding predators. Their decisions, for example, where to go or how to respond to danger, or which direction to swim, are influenced by both individual assessments and group dynamics. In calm situations, guppies rely on their preferences. However, when faced with sudden threats, panic can override these preferences, leading to more unpredictable movement. That's why we decided that we wanted to do this experiment in panic conditions or in condition of threat. For this experiment, we use this tank, which has a tunnel that ends in a funnel, and the funnel ends in an opening, in a narrow opening. This is kind of complicated, but let's just say that our goal was to make the fish move from one side to the other through this small circle in the wall. We performed 12 trials with 60 female guppies for each experimental condition. The experimental conditions were one opening to the left, one opening to the right, and 2 central openings. All treatments showed mix of clogging and burst movement that you can see here. Here, this is clogging and bursts. We can see that all of them had clogging and bursts, which affected flow. The treatment with double opening was the one that had more cloggings, but also the one that had the was the one that had more cloggings, but also the shortest time of clogging time. And the proportion of time spent clogged is this one. Was similar in all of the treatments with just some slight variation, but basically the same. We also did a power law tail proof, and we can see that the flow is basically the same in all the 3 conditions. We also did these graphs for evacuation curves. Each line represents a test, and again, it's basically the same. So with these things, with these tests, we can know that the right opening had the highest flow with 1.78 fish per second, while the left one had the lowest flow with 1.34 fish per second. However, it was not a significant difference. Um, and the double rate was 1.76 fish per second. It's important to notice that the double treatment did not surpass the right and left treatment combined, which would be a flow of 3.12 fish per second, which means the system did not benefit from the added path. And this supports the Brace paradox. Also, we thought that maybe this would have to be with lateralization. And I know there is a lot of people that's going to talk about it, and they are probably going to do a lot better a job explaining this than I am going to do. But basically, lateralization means that you prefer one side of your body over the other. In humans, we can see it because we are left-handed or right-handed. And in fish, they usually prefer to swim in one side or the other. We thought that this might explain why we had the Rice paradox. However, the laterality index was minus 0.16 for the double opening trials and 0.06 for the single opening trials. In order for it to be a lateralization, it should be close to 1, either 1 or minus 1. But this is nowhere close to 1 or minus 1. So this indicates that there is no lateralization. So to sum up, in all cases where bursts and clogging periods, the double treatment fell short of the expected efficiency based on the principle of additivity, which means that the sum of the right and left treatments was not equal to the flow on the double treatment. So the Price paradox is present in the system. There was no lateralization, suggesting that the environmental configuration had an emergent effect on the collective behavior of fish. I would like to say thanks to my advisors, Dr. Palestina Guevara Fiore and Dr. Felipe Pacheco Vázquez. I would also like to thank the team at Laboratorio de Ecología Evolutiva de la UAP, especially Eduardo López Sosa, who is one of my co-authors in the paper that we are going to publish about this work. And that's all. If you have any questions, I would be happy to answer them.
Okay, if it's not any question from some of the participants that are already enrolled in this panel, we are fast with our next opponent, Miguel Trujillo Garcia. Are you there?
Sorry, I think the next opponent is Jessica.
Oh, sorry. Let me add her. Hi, Jessica. Nice to meet you. Can you hear me? Yes.
Yes, I can hear you.
Yes. When you can upload your presentation, and I'm going to put you on stage. We pass with your talk, and I'm going to let the stage to hear you.
Okay. It, can you see it now?
No, it's not in the. No, in the part below of the screen when you are seeing me is presentar o, sorry, I don't speak Portuguese, no? Presentar o convidar. Yes, sorry, if I make them, yes, are you there? And let me leave the stage and you are ready to.
Well, hi everyone. I'm going to talk about how microplastics affect Mexican native cycles Mojarra criolla and Phyllophus islanus. First, I was going to talk about aggression, but for the data analysis, we decided to take aggression out of the project. So now I'm going to talk about feeding latency and respiratory stress. First of all, what are microplastics? Well, we start with a plastic. Any kind of plastic is okay. Here I use a water bottle for simplify this. This plastic is thrown away in the trash and usually ends in the natural environment. Here, the plastic is exposed to physical, chemical, and biological forces that breaks down the plastic into smaller and smaller pieces until it reaches the size where we can call it microplastic. Microplastics are then a small piece of plastic that are from 1 micrometer to 5 millimeters of size and can be divided into two big groups. Big microplastic that are above 5 micrometers and small microplastics that are below 5 micrometers. And these microplastics that are in the environment are now available for the organisms that live in there. I'm going to talk especially about aquatic organisms with emphasis in fish. So fish can be exposed to microplastic in two pathways. The first one is the direct pathway, that is by respiratory exposure, skin absorption, or by foragic behavior. Here, the fish can eat actively the microplastic if they are big, or accidentally if they are small. The second pathway is the indirect pathway, where the fish can eat other organisms that are already polluted with microplastics, or can transfer this microplastic through reproduction or by being preyed by higher trophic levels. Now, this microplastic, when they are big, they usually get into the guts and remain there. But here is when the things get a little interesting. The small microplastics can be distributed into the organism in the gastrointestinal membranes through endocytosis and be transported to different organs by the circulatory system. These microplastics usually get into the gills, but can also end in brain or in muscle. Finally, these microplastics are eliminated from the organism in the excretion, where small microplastics have a smaller rate of depuration than big microplastic that are eliminated faster. And finally, they can also be eliminated by skin desorption. The adverse effects on organisms by microplastic can be mechanical abrasion of blockable structures, local and systemic inflammatory responses, oxidative stress and cellular damage, or metabolic, hematological, reproductive, and behavioral alterations. Now, the organism that I'm working with, the Mojarra criolla, is native of the Balsas Basin that is in the central part of Mexico. This organism is important for the locals as they fish and eat them, and is especially vulnerable to microplastic as it has an entomophagus specialized diet, and this makes it vulnerable because insects are vectors of microplastics. So we wanted to figure out what were the effects of microplastic on the behavior and the aerobic metabolism of Mojarra criolla. For this, we will lay different variables. The swimming activity of Mojarra, its reproductive use, feeding duration, feeding latency, its oxygen consumption, and finally the amount of microplastic in feces to evaluate the depuration of the pollutant. And we did this in 3 different period of times before being exposed to microplastics to figure out the basal levels, then how it changed when they were exposed to microplastics, and finally, without microplastics to see if there was a recovery. For the experimental condition, we put every single fish in individual aquariums, and we made a microplastic solution with a concentration of 1 milligram per liter and a size of 1 to 5 micrometers microspheres. So we start the experiment with 15 days of acclimatization of our fish. Then we settle the start of the experiment with 1 day of pre-exposure, and then 2 weeks of exposure to the pollutant, and end it with 1 week of depuration where we took out the microplastics of the environment. We have 2 groups, one with microplastic and the other that remain without microplastic during all the experiment. Here we can see these dark dots in day 1, 7, 14, and 21. This marks water change, and we also use this day for the experiments for the behavior and the oxygen consumption. So one dark dot day looks like this. We first took out our fish of their aquarium and put them into respirometry systems. Then we make sample collection of the species and the water change. Then we put back the fish into the aquariums. Finally, we make videos to evaluate their behavior. Here we have the respirometry system, where we measure the routine metabolic rate using the oxygen consumption of the fish. For the amount of microplastic in feces, we took the samples, we clean the samples, we then dry them, weigh them, and count the microplastic using a stereoscope microscope. Finally, for the behavior, we did 15-minute videos where we evaluate the time of swimming activity, refood use, the feeding latency, that was the time since we put a food flake and the organisms contact the food flake, and the feeding duration, that was the time that the fish took to eat one flake. For the data analysis, we use a generalized linear mixed model and linear mixed models for the behavior and aerobic metabolism. If our model were significant, we made plant-to-net comparisons to evaluate the difference with and without microplastic group in each of its period of time, pre-exposure, exposure, and depuration, and for every single variable. Finally, we use a Kruskal-Wallis analysis for the amount of microplastic in feces. We saw that there was a difference in oxygen consumption in the exposure period of time, where fish that were exposed to microplastic have an increased oxygen consumption. And this also can be seen in the triple fin fish. And this may do because a gill obstruction with microplastics that force our fish to compensate it, increasing its respiration. For the time of swimming activity, we didn't find any difference, and it also happened for the refood use and for feeding duration. This may do because the effect on metabolism does not compromise essential activities. But for energetical demanding activities, microplastic may be making an effect, as it was seen in triple fin fish, where microplastic exposure led to a decrease in maximum swimming speed. For the feeding latency, we found a difference in the depuration period, where the organisms that were exposed to microplastic had a smaller feeding latency. And this may happen because during the exposure, the fish feed off microplastics, making them have a false sensation of satiety and a reduced nutrient absorption, making them have an energy deficit. Then during depuration, because the fish were no longer eating the microplastic, this false satiety disappeared, but the energy deficit remained. So they have an increased motivation to feed. Finally, for the amount of microplastic in feces, we didn't found any significant difference between the exposure and the duration period of time. And this may be because the microplastics were very small and we have a high concentration. So there was a high intestinal retention and low elimination rate. So we could find that Mojarra Croya had a reduced energy efficiency due to microplastic exposure, and this can increase its vulnerability to environmental stressors, especially in limited food availability context. And also we watch a persistent of microplastic in Mojarra Croya, so there is a bioaccumulation potential, putting in risk not only the Mojarra Croya, but also its predators, the natural and the human that eats this fish. Finally, I want to thank my thesis committee and PAPIT project for founding this project. And if you have any doubts, I answer there. Thank you.
There is a question in YouTube from a participant is, were you able to measure the microplastics in whale? And is there a concern about eating the fish when caught in the wheel? But two questions.
I wasn't able to measure my plastics in, sorry, I can, I wasn't able to measure the microplastic in the natural environment, but it is found that there is about 800 to 1,600 microplastics in a kilogram of sediment. It was a study that was made before. And there is not regulation for eating Mojarra Croya that I know.
Okay, there is, oh no, it's the same. Okay, I think it's not more questions, so it's okay. We can finish and pass with the next, with our next opponent. Miguel, are you there? Yes. Let me only put you on the stage.
Do you hear me?
Yes, we hear you. We hear and see you. We hear and see you.
Okay, thank you. Just give me 1 second.
Yes. Yes.
Presentar.
I think it's a little echo. Maybe you are in another screen, the YouTube. Because if not, you are going to hear your own. No? Okay. Maybe it's my phone.
Okay. Can you see my screen?
Now we can.
What about now? Hello?
Can you hear me?
Do you hear me?
Yes, we can hear you and see you. Your presentation is in the screen.
Okay, thank you very much.
Yes.
Well, hello everyone. My name is Miguel, and I'm going to present some results of my PhD thesis. The title of my talk is Failure Cannibalism in the Redhead Goby, Elacatinus puncticulatus, and First Insights. I'd like to define first what is filial cannibalism. And it's the act of eating your own progeny, as in the example that we can see in the right side. and that is a centipede, eating its young. It's the mother in this case. Filial cannibalism has been evidenced and documented in different taxons. And in this case, I'm going to talk about what happened in this small fish. And filial cannibalism can be divided into two main categories. The first is total filial cannibalism, in which parents usually eat the whole offspring or brood, and partial filial cannibalism, in which the parents eat just a part of their young or eggs. In our study, we are performing experiments on a small fish called commonly as redhead goby, and some different features characterize them. The first is, as the name suggests, the head color. In this case, we can see an orange color.
Sorry, Miguel. Someone advised me that they can see the presentation in the screen. Can you open your PowerPoint presentation, because you're having the slides, right?
Sorry, I can hear you. What?
Sí, yes. We open the presentation, like if you are presenting, maybe that's the trouble, because we see the slides in the left side. Maybe
You see the slides?
that's the reason. Yes.
You mean that you don't see the full screen?
No.
It's in full screen right now.
No, we are still seeing. No. We can see all the slides in the left side, and maybe that's the trouble, because some people advise me that they are not seeing the presentation.
Okay.
Let me only check.
I'll do it once again.
Yes. Do it again. Upload again. Okay. Let me only check.
What about now? It's in full screen.
I'm checking with the console of viewer. Let me one moment. No, they can see. They can see it. But I have you.
You can see the screen or the slides? What's
No, I can see them, but the people, some people can't.
Some people can't, but you actually see the screen? Hello? ¿Qué está pasando?
Okay, I don't know why Miguel?
Yeah.
If we can see the slides, no, there is the PowerPoint presentation and the slides in the left side. Try to upload your presentation again. If that works, we continue. Maybe it's not the best option because you are not being seen in full screen. But if not, we continue. What do you think about this?
Okay. I'll do it once
Yes.
again. From the very beginning, you mean?
Yes, do it at once. No, you only upload your presentation is the screen that we are presenting, and over is apresentar or convidar. You press in that and share your presentation. No, not from the beginning. I don't know if you as you feel comfortable. If you want to start from the beginning, it's okay. If not, since the point that you left.
Okay.
Okay.
What about now? Do you see the whole screen or still the problem persists?
The problem persists. We see the orange part and the PowerPoint presentation, but we can see the full screen.
Hmm. Okay. I don't know what's happening.
Yeah, some people, some participants put you in the
Yeah.
private chat. Some suggestions. For example, the Yanira says, put the button present. No, put the button present to full screen, and that's all.
Sí, sí,
But
sí.
I don't know if yes. Yes.
What about now?
That's the yes. Yes.
Okay.
Solve it.
Okay, well, I was saying that my study species is the redhead goby, and some characteristics are present in this species. As I previously said, the orange color on the head, or in some other populations, the color of the head is red, um, and they possess these neon lines over the head, an orange line and a blue line, and they also possess some black spots across the body. And there are small species up to 5 centimeters long. And the scientific name is Elacatinus punticulatus, and this is the species that we are using in the laboratory. Where the organisms come from, where the study was performed in Mexico, and this species is distributed along the coast, starting in the Gulf of California, all across the coast to Panama. In this case, we capture individuals from La Paz in a place called Puerto Mejia. By scuba diving, individuals were captured using hand nets and then transported to Sisimar IPN. All the individuals captured were maintained in a stock tank. This stock tank was maintained for a few days to acclimatization and prophylactic treatments. Once completed the previous process, individuals were identified as males or females by observing the genital papilla. Following Miller, this is a structure that all individuals possess, and it's at the base of the anal fin, as the photos show. In this case, for example, the male possesses a conical shaped genital papilla, and the female possesses a wider papilla and with these protuberances on the tip. Individuals were also measured, and the standard length with a digital vernier to the nearest millimeter. In every single case, the males were also bigger than females. And this was the procedure that we tried to maintain all across our reproductive pairs. This is a photograph of how aquarium was at the laboratory. And in every single case, each aquarium poses a PVC pipe that was a nesting site and a male that was always inside the PVC pipe or near to the PVC pipe, that is the nest, and a female that was always outside the nest, or almost always. Reproductive pairs were maintained under specific characteristics, such as the photoperiod was maintained at 16 hours light and 8 hours dark, salinity was maintained at 36 ops, and temperature ranged from 25 to 27 degrees, and the feeding was twice a day, one at 10 a.m. and one at 4 p.m. with adult Artemia. It's important to say that these results are not specific experiments performed to evaluate filial cannibalism. These experiments were performed to evaluate parental care behavior in this species, and performing these experiments, we found filial cannibalism as a side project. Well, 2 experiments were used to present these results: 5 replicates using just 1 nest and 7 replicates using 2 nests. The nest was a PVC pipe, as I previously said, and with a diameter of 2 centimeters. In the first experiment, we tried to count the number of eggs per day of development. I mean, from the day of spawning until a day before hatching, in which we evaluate 5 replicates with just 1 nest. Each nest contained a plastic sheet and rolled inside the nest. And this was used to take some pictures on the spawns, as we can see in the right side of this slide. From these photographs, we were able to count the number of eggs every single day, and we see in some cases a reduction in the number of eggs, as we can see here in the day 1 compared to the day 3 of development. The day 1 is the day of spawning. This was the first incidental result of filial cannibalism. We also calculated the filial cannibalism percentage by each day of development, and it ranged from 1% to 100%. And as I previously said, pictures were taken from every single day of development, from the day 1, which was the day of spawning, until day 5. And in total filial cannibalism, I mean the whole consumption of the eggs, was performed during the first 3 days of development. The mean percentage of cannibalism on day 2 was 75, while for day 3 was 66. In comparison to partial filial cannibalism, the percentage of cannibalism was lower. The higher rate was for day 5 to day 6 with 28%, and from day 2 to day 3, 11%. Up to this point, we know that this species perform filial cannibalism, but there's another question. Which sex is performing this cannibalism? The male, the female, or both parents? Well, this question was partially solved with another experiment. We were performing another experiment filming males, parental care in males, and these observations were every single day of development of the eggs, and we were able to see the activity of the male in comparison with the eggs and with the nest. And in some videos, we were able to see the females cannibalizing on eggs, and one replicate using just one nest and one replicate in two nests. As this, in this case, the female enters into the nest while the male is outside the nest and starts to bite the eggs in an abrupt manner, as in this video. What's happening? Uh-oh. Do you hear me?
Yes, we can hear you. The video freeze or only the or all the presentation.
What? Sorry.
Well, the video freeze or all the presentation.
Oh, okay.
All the presentation. Only restart the program. Maybe we are going to lose the image, but you only need to reconnect again in the same slide.
Okay.
If happen again, is the video.
So I skip the video.
Look, try to reproduce it.
Do you see it?
Yes, we can see it. We can show it.
Okay. Thank you. Well, as I was saying previously, in this case, we were comparing how this behavior in females was in comparison to males. We were able to evaluate and distinguish parental care behaviors in male. In this case, these species perform male-only care. And as we can see in the right side, the male is touching the eggs in a gentle manner. In contrast to the left side, the female just bites the whole clutch in an indiscriminate way. We also were capable to see that females also consume the larva in 3 replicates out of 5, in which we use on a single nest. While the male released the larva from the nest, the female consumed them, as we can see in this video. But what about the male cannibalism? I did a more recent experiment in which I was thinking that I would be capable to calculate the percentage of fertilization of the eggs. So what I did, just separate the eggs from the day of spawning, and every single day I was taking pictures, but these spawnings were separated from the parents, so I was avoiding filial cannibalism. But in this case, I was able to observe that some of the eggs were dying. I'm not sure if in this picture you can see that some of the eggs present 2 black spots, and this means that these eggs are developed, and some of them are white or yellowish. And as we can see in this figure of the right side, the eggs with eyes were developed, and the white eggs were infected with a fungal infection. In this case, after observing this, I decided to return the whole spawn to the male parent and take another picture a day after. From the day 4 to the day 5, the male was capable to consume part of the clutch, but the eggs consumed were the dead eggs. And this is supported by another hypothesis that explains the origin of filial cannibalism, in which states that parents usually consume progeny that is dead or infected. And in this case, we suppose that this kind of cannibalism is performed by the male only because males spend most of their time inside the nest. And this behavior is observed in a wide range of species, in which males spend most of their time, from 80% to 90%, inside or patrolling the nest. And another reason to speculate that this cannibalism was performed by the male is just that inspection is believed to function as a cleaning behavior. Inspection is a behavior that we identify in a previous paper, and in which males usually observe the eggs, and in some occasions they start to bite them. And as conclusions, in this case, we can see that females consume eggs and larvae, apparently in an indiscriminate way, and males consume dead eggs, but some additional experiments are needed to clarify this situation. And an undergraduate student is going to perform another analysis in this sense, and he's going to evaluate some other hypotheses on filial cannibalism or males. Thank you very much.
Thank you very much, Miguel. I only have one question.
Yeah.
What do you expect if you stop males and females to eat the eggs? And what is going to develop the eggs in both hypotheses?
I didn't get your question, sorry.
Okay. Imagine that you the fathers put the eggs, no? They opposite. And you remove the fathers and let the eggs develop until adults. What do you expect
Okay, you mean
that scenario?
Okay. This is the case in which it's a difficult speculation question, because I separated the eggs from the parents in this last experiment, and the eggs started to death. And in some occasions, they were infected by a fungal infection or bacterial infection. And in some cases, the whole clutch dead. So none of them hatched. So that means that when parents are present when male parent is present, it's really important for the survival of the hatching. I think if you left the whole clutch alone, I mean, in laboratory, it's usually infested. And in the wild, usually it's not infested, but it's consumed by conspecifics or heterospecifics.
Okay, thank you. We have also a question from YouTube. Let me read. What are the ethical implications for this kind of experiment? Considering that this experiment starts as parental care.
You mean?
In other words, the species depends on parental care.
Yeah,
if
up
you
to some
remove the
point.
parental, okay, sorry.
Yeah, I understand. If I remove the parents, is that the question?
Yes, the ethical implications of removing the parents.
You mean that it's not ethical to remove the parents, or I'm missing the question? I'm not sure if I understand.
Yes, for example,
Well,
it's a species that present parental care. If you try to prove some stuffs in this species, what is the limit of do it to remove the parents in the ethical dimension?
okay. I just saw that there are different, in the journals there are different guidelines. So you can see that some inhuman experiments are prohibited, and in this case, I don't know. It's just that it's a new topic for me. It's a question that I cannot answer completely right now because uh, this species is important in the aquarium trade, and some results in this sense, I think, are important for production, for example. But I think that it's necessary to know if you can develop the whole clutch with parents and without parents, because when you are producing different species, you're looking for to reduce the cost. In this case, I was trying to find some other results, but I found the filial cannibalism. The ethical implications in this case, I can not answer the question right now because I don't know which would be the possibilities in this sense. Sorry. Uh,
Well, thank you for your answers.
okay, thank you.
Hi, Lupin, you're on the stage.
Hi everyone, can you please put my poster up on the screen?
Wait.
Okay, can you hear me just fine?
Yes.
Okay, thank
Can
you
you
so much.
hear us?
Yep.
Okay. Do you start?
Good afternoon, or good night, or good morning, everyone, wherever you're from. My name is Lupin Telles, and today I'm going to present my research named Ambidextrous Squirrels, Environmental Factors, Not Laterality, Predict Pulp Preference in California Ground Squirrels. To start, behavioral laterality is found in many animal species, including domesticated chickens, dogs, guppies, and also some types of monkeys. And what is laterality? And basically, it can be explained by specialization of the brain's hemispheric. So, and this specialization results in a asymmetric limb preference, just like us when we have a more preference for right hand or left hand. And there's also evidence for laterality in a lot of squirrel species, just like gray squirrels, pallas squirrels, and also date rock squirrels. And in all of these tests, to assess this laterality in them, they were presented a task that included a cylinder, and they had to reach for the food that was inside the cylinder. And while they would do that, only one of their paws could be fitted in. And the results that came was that some of them had a preference for the left paw, but most of them actually had no preference at all. So this research we wanted to assess if California ground squirrels, if their paw preference could be best explained by species-specific laterality. With this, we would see a preference for each of their paws in a population level, or due to external factors. For the methods, we used a population of individually marked California ground squirrels at Briones Regional Park in California, and we showed them a fully automated lateralized lever puzzle where we had a horizontal lever that could be pushed either to the left or to the right. With this, we had 3 boxes and totalizing 35 hours of video recording, because we are also recording them. After that, we scored the videos using the platform Boris, where during our score, we would tell if they used the right paw versus the left paw and which lever side they pressed. We also used a binomial generalized linear mixed model to test if the paw preference was predicted by the lever side with the squirrel identity as a random effect. In other words, the identity would not affect the choices. And to assess this potential individual laterality, we also calculated the individual level of repeatability in paw use, if they used more one paw than the other. And the results that came was that in total, 36 squirrels of that population solved the puzzle. And by solved, we mean that they pressed the lever in each of the sides, produced, and in total we had 592 solves using the right paw and 612 using the left paw. The right lever was pushed 471 times, while the left one was pushed 733 times. As said before, the lever side was a significant predictor of the paw use with a p-value of less than 0.001. And as you can see here in our graph, when pressing the left lever, the squirrels used more of their left paw than the right paw. And when using the right lever, the same results came. They used the right paw more than their left paw. So we had a 75% of results of using the left paw with the left lever. And with the right lever, we had a 93.7% of usage of the right paw. And as for the repeatability, if each squirrel used more one paw than the other, the results were actually negative. We found no evidence for repeatability, and for that we used a bootstrap method, where we bootstrapped 100 times to get this result. And as for the discussion, as said before, we found evidence for behavioral flexibility rather than laterality. In other words, they actually adapt to the external factors to use each of their paws. And our findings also show that individuals, as they're adjusting, this can also mean that there are other studies with mice that showed that for less complicated tasks, they use each of their paws, but when these tasks are more complicated, they actually chose one over the other. So this test shows that they are indeed flexible they were flexible for this test, but it doesn't mean that they don't have a preference if they are showed a more complex puzzle. And so we should emphasize the importance of carefully considering the nature of manipulative tasks and also the environmental factors when we are going to evaluate laterality in animals. I like to thank you for all the funding that was necessary for this research to be made, for my co-authors, and also for the ISCABC team for all of this event. Thank you all for watching, and let's hear some questions.
Someone or can join to the conversation to give some questions or a comment. We can wait a few. Okay, meanwhile, I want to ask a question. For example, we imagine an idealistic population. What implies, for example, that more than 5% of population has laterality to the left, no? Maybe in the squirrels, no? But in general, not only with the squirrels, with other rodents, what implies that the means was over a laterality of the right or the left?
Oh, you're asking if they had a laterality level?
Yeah, no, I imagine, for example, you have upper 50% of the population has a laterality to the left or the right. What are the implications of that for the population?
That basically just means that their brains has a hemispheric specialization, and that their brain in each of the sides they use more of their brain to certain tasks, while the other hemisphere is not that much used. Did I answer your question?
Yes, thank you. And question YouTube. Can you hear me?
Yes.
Because I lost my. I think. Okay, I appear in the screen. So, okay, says lateralization depends of the experiment design. Meaning, do you think if we change something in experiment design, your results could change?
Yes, as I said by the end of my discussion, if we change the environmental factors, maybe the results will change. For example, as I said, of the research that was done with mice, when they were presented a more complicated tasks, they would have a preference of limb over the other, while on everyday behaviors like grooming and self-cleaning, they would interchangeably use both. So if we for these puzzle boxes, we find that it was a pretty easy task, as they only needed to press with their body weight to solve the box, and it would deliver them some seeds and some food. And if we present a more complicated task where they have to think about it and how to open and how to solve it, maybe they would have a preference, or maybe not. We need more research on that to be sure.
Well, thank you for your time and for your answers.
Thank you so much.
Oh, sorry, we have a last question from Catarina Villa. Says, great work. Did you also test repeatability of how use only on left lever, for example?
No, we didn't test. The repeatability test was only done on individual levels. So we only test the repeatability for each of their paws for each squirrel. So it was on if they chose one paw over the other, not if they use more the right or the left. Does that answer your question?
She don't reply, we are going to give her a minute, no, to reply if she wants. I think not. I mean, I think I understand that you answer her questions. If there are, okay, no, there is a reply. This is a little bit lag. Says, yes, but I was thinking that maybe they do show repeatability, within few flexibility of using both paws.
Yeah, this maybe it is, but with the research that we did and the numbers that we have, at least in this population, they didn't show enough. We didn't have enough data to confirm this, basically.
Okay, the same, we're going to give her a minute to reply or make another question. Say thanks.
Thank you so much.
Meanwhile, we are going. Okay. Meanwhile, we are going to say goodbye only for today. We're going to see again tomorrow. If you have any questions, you can write us to the mail, or you can find in the page, and that's all. That's for all. That's all for now, and see you tomorrow. Have a good afternoon, whatever you want. Bye, everyone.